So today we're going to be looking at chromosome abnormalities called aberrations, specifically inversions. So here we're given two chromosomes where each letter represents a region on the chromosome and the dot represents the centromere of the chromosome. If it is in alphabetical order like the first one is, that means that there are no abnormalities. So first we're going to look for similarities in the regions of chromosome. If we flip the second one, then we can see that the only part that's different is the middle, and the ends are the same, as represented by the green lines. So the light green arrow shows the direction that it should be going, alphabetical I, J, K, and the inverted one is K, J, I. So it's inverted because the part of the chromosome is flipped around, and since it involves the centromere, the dot, we'd call it a pericentric inversion. So whenever we have one normal and one inverted chromosome, there's always going to be problems when they try to line up. As you can see in the middle, the centromeres don't line up, and neither do the regions K and I. So in order to have maximum pairing, we have to form what's called a compensation loop. So in drawing a compensation loop, first you're going to draw your normal chromosome, and it's going to have a big bump in the center, and then you'll draw your chromosome with the inversion and do a loop around inside of it. So each line here will represent a chromatid, and the pairs of lines will represent a chromosome. So now that we have the compensation loop, it allows all regions to line up, including the centromeres, and we're told that a crossover event occurs between J and K. So during meiosis, whenever a crossover event occurs inside of a compensation loop, it can create inviable gametes. So we're going to go through each chromatid and see if that would create a viable gamete due to the crossover. So we're going to start with this top chromatid. If we trace it, it has no change because it's not involved in the crossover, and since it's unchanged, we're going to call it a parental. So we'll follow the next chromatid and we'll get to the crossover and go to the loop it's crossing over into and keep following it along. And then we'll write down what that gives us, and this will be a recombinant type because it was affected by the crossover. So we do the same thing for the next chromatid. It's going to be parental type as well because it is not affected by the crossover. Then we have to come around to the other side because we've already used all the left-hand ends, and we're going to do our last chromatid. So we go over to the crossover and we just keep on tracing it, and it's another recombinant type because it is affected by the crossover. So now we have to determine which of these chromatids would or would not create a viable gamete. Well, if it has a repeated or missing strand or region of chromosome, then it's not going to be viable because it's missing genetic information. So we're giving two more chromosomes, and we have to do the same process to see if it produces viable gametes. We can see that neither of them are in alphabetical order. They're not correct like the one in the green box. So we're seeing an individual that is homozygous for a pericentric inversion, and that both of its chromosomes have this inversion. So we're told that a crossover event occurs between J and K, and we're going to go through the same process that we did before. So since the individual is homozygous for the inversion, we did not have to form a compensation loop, which means that we are not missing or repeating any of the regions, so all of the daughter cells are going to be viable.